everyone, this is part 2 of the May-June paper of paper 1 of variant 1 of 2025. Latest one, let's discuss the other MCQs. We have discussed the 14 one in the previous video. You can watch that. Let's discuss the MCQs from 15. Which statement about the mode of action of enzymes are correct? Let's say what are this, how they are arranged, or how they are specific to their function. So number one is some enzymes have a region on their surface to which the substrate has a complementary shape. So they are talking about the active site. So we say yes, it is. Number two, so this one is correct. It's active site. Number two, some enzymes and their substrate can change shape slightly. Yes, we have learned this in induced fit model that the active site is not a rigid one. It can slightly change the shape to fit in the substrate. That's why the lock and key model was rejected and the induced fit model was accepted. The enzyme permanently change shape when they react with their substrate. No, they can go back to that change, as we discussed in 2 that they can change shape, so they can go back. It's not a permanent change. So third one is not correct. So the correct answer is 1 and 2 only. The next one is the graph shows the effect of increasing substrate concentration on the rate of reaction of two enzymes that catalyze reactions with the same substrate. So we say the enzyme P is definitely more appropriate for the substrate as compared to enzyme Q. Okay, so which statement is correct? Let's go into the day. Enzyme Q has a higher Km and a higher affinity sorry, for the substrate than enzyme P. No, it's not correct. Enzyme Q has a higher Km and low affinity. It doesn't have a high Km. First thing. No, it doesn't have high Km. So these two ones are rejected. C1, enzyme Q has a lower Km. Yes, it has a lower Km. That's why it's not reacting with enzyme Q and have a more, sorry, and have a higher affinity for substrate at enzyme P. Yes, this one is correct. That's why there is more reaction shown with enzyme P as compared to Q. So Q is showing the lesser, you can say potential. It's not reacting to that much extent, but the P1 is. The cell surface membrane structure is described as the fluid mosaic model. Yes, because it's made up of phospholipid bilayer. So we say there are two layers and there are some proteins. Let's say there are two layers and there are some proteins that are embedded in it. So it's a fluid mosaic model. So what correctly describe the mosaic? So I've told you that there are certain molecules that are embedded in it. So already mentioned what structures are these. The different patterns that are obtained, no, phospholipid molecule is not going to help. This one, this proteins are going to help. The random distribution of cholesterol, no. The regular pattern produced by the phospholipid head, no. The scattering of different proteins, yes. This one is the answer for this one, the fluid mosaic model. So a student immersed a plant tissue in solution S. Let's say we have S solution. For the next 30 minutes, the student used a microscope to observe the cell in the plant tissue. During this time, the cytoplasm of the cell shrunk. It means the water moved out of the cell. Let's say water move out of the cell and longer pushed against the cell wall. In many cells, a space appeared between the cytoplasm and the cell wall. So there is a gap definitely because the cytoplasm shrunk. So which statements are correct? Number one, the water potential in solution S was more negative. Definitely there is lesser water in S. That's why water is moving out. So this one is correct. Then the water potential of the cytoplasm at the start of experiment. Without a cell wall to provide support, it is likely that many of the cell would have burst. No, the cells are not going to burst because its cell wall is much stronger than cell membrane. The clear space between the cell wall and the cytoplasm of many cells was filled with air. No, there is no air in that. So the only correct option is 1. Let's move to the next one. A sample of 500 cells dividing by mitosis was examined to identify the stage of mitosis for each cell. Okay, so we are on like cell division. Let's see which phases are this. Number one, the late prophase, mean there are chromosomes and the cell prepare itself for the cell division. So here the preparation is taking place. Number two is end of metaphase, so they are aligned on equator. The last one is between the start of anaphase and the end of telophase, means that they are going to get separate. The sister chromatids are going to get separation. I'm writing SC for sister chromatids. So which row is correct? Number one, the number of cells at the equator, so it is 45. So let's say 45% of 500. So it would be like 45% divided by 100. Whenever there is percent, you need to divide it with 100. Of mean multiply of 500. So the answer would be 225. The second one is where the number of cells with separated chromatids. So here is the separation take place. I've already mentioned. So 24 divided by 100 multiplied by 500 is 120. So the answer is A. So you just need to know about the basic calculations to solve this, and many students got it wrong because they cannot just identify the stage where this process is actually happening. So here we have a chromosome that we have discussed earlier, that in the mitosis the cell are going to get separate. So we have chromosomes. Let's say how they are going to act. Number one, what are the structures P and Q? So P is this very central part. It's a centromere, and Q is the arm which is going to cut down to prevent the loss of any important information, and they are present on each one of these. So the answer is telomere. Answer is B. So what is the importance of telomere? So there is an interesting video that aging is also related with this. You need to watch that video, go back to my channel and look at the significance of this telomere. So which process are used by the stem cell during tissue repair? Sorry, which processes are used? So in tissue repair, the cell has to be formed. So the tissue is a group of cells. If the group of cells are damaged, then new cells needs to be formed. Let's go into the detail what is needed. So very first thing needed is the protein that we usually believe helps in the repair of cells. So let's go into the 21. What do we need? We need cytokinesis. Yes. So we need cell division. We need DNA replication. Of course, the mitosis is taking place. And we need transcription. Yes. The DNA has to be converted into mRNA, and that mRNA then make proteins. So all these three processes are equally important. The next one is the enzyme telomerase prevent loss of telomeres after many mitotic cell cycles, as we discuss here. That this is very important part. It is required so that the extra information is not so very important information is not extra, is not erased. So which cell transcribe the high concentration of telomerase? Number one, the neutrophils. Number two, the matured red blood cells. Of course not. Then the activated memory T lymphocytes. Yes. So the answer is 3. Not the neutrophils, not the cell, but the T lymphocytes. So the next one says which statements are correct. Let's say all polypeptide are coded for by genes. So let's say yes. Next one, all genes are made up of sequence of nucleotides. Yes, there is a proper sequence. All gene mutation result in the change in nucleotide sequence. Of course, that's why it is mutation. All gene mutations will result in altered polypeptide. No, because sometimes it is missed, or sometimes it is added or skipped. So this can be incorrect. So all these three, the above three, are correct. So the answer is 1, 2 and 3. So a double-stranded DNA molecule was analyzed and 29 of its nucleotide bases were found to be adenine. Let's say we have 4: A, T, G and C. Be very careful. The A is actually paired with T and G is paired with C. So if we have, how many A do we have? We have 29 A. So definitely we are going to have 29% of T. So collectively it makes 58. So what is left? We are left with only 42. That is collected. So if we divide it with 2, 21% is the half of this. So how what percentage of cytosine is present? That is 21%. So in some cells, the non-coding sequence of RNA, so there are some, are removed after transcription. Which row correctly state the name of these non-coding sequences? So the non-coding one are known as that we are going to learn, and the type of cells in which the non-coding sequences are removed. Okay, so the name of the sequence of non-coding sequence, that is intron, and the type of cell in which it is removed, that is eukaryotic. So the correct option seems to be C. Let's move to the next one, 26 one. Water potential are measured inside a leaf cell, in the air spaces of the leaves, and in the atmosphere. Okay, so we are going to calculate both. The psi sign is for like water potential. So what value would allow the water to move from leaf to the atmosphere? So if there is lesser water outside, lesser water potential in the air, only then water is going to move outside. So first of all, the inside cell we have -1, and outside this the airspace. Okay, this is airspace and this is outside. So here we say there is much lower water potential outside, that is -100. So more negative value mean more smaller value. So water will move out of the cell. So without moving to the next one, sorry, we'll choose this option A. Next one. The sample of liquid are taken from the different locations in the plant and tested to measure the concentration of four solute, that is sucrose, nitrate, amino acids, and magnesium. So which samples have been taken from phloem seep tube uh, elements? So we say that if we talk about the phloem, here we have two things. One is sucrose and other is amino acid. So these two are transported through phloem. So which option can be correct? Sucrose is this one. Very high sucrose here and very high sucrose here. Very high amino acid here and very high amino acid here. So the option is 1 and 3. Simple. You just need to recall the basic you can say concept. So to is it number 2, a heated band was wrapped around the stem of a plant at a height of 1 meter above the ground and left for 10 minutes. So it's not that much high. So how will heating a small section of stem to 60 degrees Celsius, you are going to affect the different processes, the transport of phloem sap and the xylem sap between the roots and leaves. So what is going to affect? Definitely the xylem is going to affect first thing, because it's taking water from ground till upstairs, to the upstairs I'm saying upstairs to the up to the leaf. So this is not going to be affected at all, because water has much higher specific heat. And the second one, what is going to stop, that needs to be the transport of phloem sap, because this is going to be affected. So this is all for this video. To watch the solution of other MCQs, you need to switch to another video.